Well, welcome everyone to another session of Conversations with McGill OSS. That's the Office for Science and Society. I'm Joe Schwartz, I direct this office, and with me today is my colleague, Jonathan Jerry. And our usual complement of Emily Shore is not with us today because she happens to be moving. So we did give her the day off. But our special guest today is Dr. Jonathan Kimmelman, who's a McGill professor, James McGill professor, which is a special kind of professorship. And he's a professor of bioethics. Of course, he is very interested in the whole vaccination business, which will be focus of our discussion today. So welcome, Jonathan. And just to get started here, what does a professor of bioethics do?
Okay, well, so, there are any number of different ethical issues that come up in medicine, that come up in drug development. For example, is it ethical to deliberately infect people with viruses in order to test vaccines? Is it ethical to withdraw life support once a patient has reached, a terminal stage of disease? Are practices like medical aid in dying ethical? These are the range of different kinds of ethical issues that come up in medicine and life science research. And what we do is spend a lot of our time doing research and thinking about the conditions under which those kinds of practices can be ethical and conditions under which they can't be ethical. So basically, many of us spend a lot of time just sitting and thinking really, really carefully. But also many of us do a lot of research because a lot of our judgments about what is ethical and what isn't ethical will often turn on empirical findings, on evidence. And so a lot of times we spend our research efforts trying to gather evidence to help us to support decisions that are going to promote the kinds of ethical priorities that we have in medicine.
Well, obviously, these days, everybody out there is talking about vaccines, as if this is going to be the savior. And a lot of people have the impression that this is going to be sort of like a light switch it on and our problems are going to disappear. That, of course, is not the way that this is going to play out. So we'll start at the beginning. I mean, we've had over the last 2 weeks, a lot of very interesting and certainly information that does sort of give us a bit of a glimmer of light at the end of the tunnel. So it is somewhat encouraging. But as is often the case, people take a small step, and by the time the press interprets it, it is as if a giant leap has been taken. So let's discuss these 3 vaccines that have come out. And I guess before we go any further, we should talk a little bit about the difference between these vaccines. So Jonathan, maybe you can start with that and just differentiate, what the differences are between these vaccines.
Sure. I mean, I can say a couple things. There are a lot of different platforms or a lot of different strategies one can use to develop a vaccine. The 3 vaccines that we've probably heard the most about use one of 2 different technologies. One of them is that they take a virus and genetically modify a virus that normally causes common cold, to express a protein that triggers a really potent immune response. And so that's the AstraZeneca technique or strategy. The other strategy that we've heard a lot about, namely the Pfizer and Moderna strategy, is using mRNA. So mRNA stands for messenger RNA. So it's kind of, you go from a gene, which is in DNA, to mRNA. And then mRNA is then used to produce proteins. And the proteins that those mRNAs produces also trigger really, really potent immune responses. So those are the 2 big platforms that have, at least at this point, seem to have reached pretty close to the finish line in terms of completing large field clinical trials.
Now, the messenger RNA vaccines, this is a platform that has never been used in humans before. So people do.
Well, to be clear, it's been tested in humans before. It's just that there has never been a licensed vaccine based on mRNA technology.
Right. So people, of course, are concerned that this is, novel technology, and by the time that millions of people are exposed to this, something might crop up that you don't expect. So the other Jonathan, Jerry, you've heard, like all of us have heard, concerns about the RNA being put into our cells and interfering with, how our body functions by incorporating itself into our DNA. How realistic is that?
It's not realistic at all. I mean, it is not something that you can just say, oh, it's a complete impossibility. But the odds of this happening are extremely, extremely low for a variety of reasons. And as was pointed out by the other Jonathan on the stream, yeah, these RNA vaccines are not new. It's just that we have never, not one of them has ever been licensed so far, but we have about 20 years of experience experimenting with these kinds of vaccines. So this RNA, first of all, our cells are filled with messenger RNA molecules. This is how life works, right? So you have the DNA, and then that DNA is transcribed into these small RNA molecules that are very unstable. They're very easily destroyed, but they act as a sort of like a paper copy of, like a blueprint for an architect to build something. And so our cells already have these types of RNA molecules in them, and our DNA is inside the nucleus of our cells. You can think of it as like the yolk in an egg. And so the vaccine, these RNA vaccines, like the one from Pfizer, the one from Moderna, what happens is that this RNA that is essentially the instruction to make the spike protein from the coronavirus, this RNA, which is in a little vehicle, like a little fat particle, for example, gets taken up in the white of the egg of our cell, so what is called the cytoplasm, where we already have a ton of these RNA molecules. It can't go inside the nucleus or the yolk where our DNA is. And even if it did, it can't simply integrate into our genome. It requires, you need certain proteins to do this, certain sequences to do this. So there's no indication so far that this could realistically occur. So this is not really a concern. And so, but again, and we've seen this with the whole GMO story. I mean, whenever you mention genetics or gene or DNA or RNA, most people don't really know the context in which these molecules exist. And you automatically go to the worst thing you can imagine is like, oh my God, it's going to turn me into a GMO. These vaccines are not going to turn us into GMOs. The real question is, will they be effective? And the preliminary results are that they are, which is fantastic. And will they be safe in the sense of, will we have some kind of immune reaction to them? And so far, again, from what has been released, it looks like they are very, very well tolerated in the short term, at least.
Okay, Dr. Kimmelman, I mean, obviously, one of the focuses of your work is to look at research and see whether it's properly carried out, and have the peer-reviewed ideas being properly followed, etc. And I know that you have written some stuff about this, and you have some concerns that maybe some of this stuff was too fast-tracked.
Okay, so I think it's helpful to appreciate that there's a lot of different research activities that are being undertaken to address the SARS-CoV-2 pandemic. And it's important not to paint with too broad a brush. So early on in the pandemic, there were a lot of clinical trials that were launched without really a solid rationale, that were launched using designs that were suboptimal at best and potentially misinformative at worst. There was also a lot of research that was launched that was not really well coordinated. So you had clinical trials trying to compete with each other to recruit patients. So there still is that kind of activity, going on, and I can talk about that. But mixed in with that research is also a lot of really, really high quality research, both for therapeutics as well as for vaccines. Now, my sense, I haven't made a really careful study of it, but my sense is that the large scale field studies, vaccination field studies, are being done with impeccable standards, in the sense that there's just an incredible amount of regulatory and public scrutiny on those clinical trials. It's important to remember that the companies running those trials want to sell their product and they're not going to sell their product effectively if people have doubts or skepticism about the integrity of those trials. So they have really strong financial motivations in terms of preserving their credibility to do the science right. And all indications I have so far, at least for the late stage clinical trials, is that these trials had a appropriate design. The statisticians that I follow on Twitter have basically said good things about the design and the analysis. And you can bet that regulatory authorities around the world are picking through those data really, really carefully. So I'm not as much concerned about that kind of vaccination research as I have been about a lot of the therapeutic research, especially that was going on early on during the pandemic. Now, there is the issue of speed. And I think here again, I can't give you a simple kind of soundbite. I think it's a really complex picture. I think that in order to evaluate the safety of a product, you need to expose a really, really large population over a very long period of time so that you can detect safety events. It's still really early on in terms of our experience with these vaccines, and it may take more time to begin to appreciate some of the limitations of these vaccines. It's quite possible that they work great in certain populations, but in other populations not so well. They may have greater risks in other populations. And that kind of textured, nuanced picture about the properties of the vaccines is going to take time. And what's also important to remember with vaccine trials is that in order to really get good signal in these clinical trials, you have to have a lot of cases. You need to accumulate a lot of cases of people developing COVID-19 in the placebo arm, typically, ideally. And so you just sort of, it's kind of a waiting game. There's no way to kind of speed that process up. And so, I think that the companies have done a really laudable job moving the pace along as quickly as possible. They've recruited really, really quickly. They've done analyses really quickly. But I think we should bear in mind that we've only seen, preliminary results for the most part, and that with time, some of the jaw-dropping effects that we're seeing in these early announcements may be a little less jaw-dropping. They probably are going to be still impressive, but a little less, impressive as we see more events accumulate and as we get more information about the safety.
There's a confounding factor in the AstraZeneca vaccine, in that this is a 2-dose vaccine. And for some reason, which seems to have been an accident, about 2,800 in the subject group were given first a half dose and then a full dose instead of the 2 doses. And it turned out that the half dose, full dose group had a 90% efficacy, whereas the others had a 62%. And this seems to fly in the face of a lot of standard science. That the dose response curve here seems to be somewhat confusing. What do you make of this?
Well, I have a couple different thoughts. I mean, the first thing to point out is that clinical trials are unbelievably difficult to do. Like every time you pick up a major medical journal and you see a clinical trial, it's a miracle they managed to pull this off. I mean, getting patients to volunteer their bodies for taking something that's unproven and, presenting their bodies to observe them over a long period of time is incredibly difficult to do. You can't cage human beings like mice and make them do all the things you want to do. And so one thing that people probably often don't understand is that, or might not appreciate, is that mistakes happen in clinical trials all the time. And in fact, this is a real sort of a research interest of my own. Just like in medicine, when sometimes people get the wrong medications, the wrong diagnoses, et cetera, this happens in research as well. It's just really, really hard to pull this off. And so it's not a common occurrence, but it's not super, super rare. And so here we have an episode where there seems to have been a dosing error early on. So I think there are a couple, things to bear in mind about this. First of all, in terms of the low dose having more of a response than the high dose, this happens from time to time in medicine. It tends to happen, my sense is, even more commonly with immune-based, therapies. The immune system is really, really complicated and doesn't necessarily work the same way that in terms of drug response that a lot of other systems in our body works, where the higher the dose you give, the more potent the response you get. So it's not, it's not totally, counterintuitive that you would have a lower dose responding. But the important thing to remember is that the trial was not primarily designed to test 2 doses. And what's more, also, that lower dose group was a younger age cohort. Those were patients that were under 50, whereas the broader population was older people, in a much broader age range. So there are a number of wrinkles in their data that should lead us to be a little bit cautious about how to interpret the results. I also think, I might have misread this, but I think Astra also combined the results of 2 independent clinical trials in their press release, which is a little bit strange. You would normally report each one of those separately. So there's a lot that is a little hazy about exactly what they showed with their result. And I think Astra, and Oxford kind of botched the public presentation. They really did not think through carefully how to go forward to the public with these results.
This is one of the problems with science by press release, is that you sort of get the bottom line, but you don't get the details. And in science, as we often say, the devil is in the details. That's what we need to really tease out to make sure that there are no surprises that are going to come. But this half dose thing, this may have been a serendipitous discovery. This works better. So it's interesting. But
let me say a couple of things really quickly. So it might be serendipitous, or it might just go away. It might regress as we get more data. So we'll just have to see. But I do want to say a couple of things about the science by press release. I mean, I think I am appalled at the recurrent use of press releases as a means to disseminate research findings. And I think that one thing that is the most disturbing, the reason why press releases are problematic as a means of disseminating findings is that they're not peer reviewed. They only provide selective representation of the data in a clinical trial. And given that sponsors are the ones that write press releases, that is likely to be somewhat tilted or biased towards what the sponsor wants to present to the public. And so, in the context of a public health crisis, releasing a little bit of information that might or might not be biased really can confound the public health response. And I think it's really kind of telling and deeply problematic that we have a legal system that puts greater priority on disclosure of information for the sake of security and exchange, and for the sake of of investment, over the priority of disclosing information in a manner that is appropriate to a public health response. And I'll just say one additional thing about this. There is a way that we could correct this fairly easily. We don't need any special legal mechanisms. Anytime you run a clinical trial, you would have to register that in a database before the trial starts. The most important and most significant database is called clinicaltrials.gov. And in clinicaltrials.gov, there is also a mechanism through which you can deposit the results. So once you complete the study, I can't think of any reason why companies, when they do a press release, why they wouldn't deposit the results of all of the top line analyses and secondary analyses that were registered in a clinicaltrials.gov registration record. Presumably, if they're issuing a press release, they've done those analyses. Why not post them at the same time as a press release?
Yeah. I mean, so often in history of science, we've seen optimism slowly fade when something new is introduced, right? And we're seeing this now too with remdesivir. At first, there was all that excitement about remdesivir. Now, all of a sudden, it's not being recommended anymore. And in the beginning, there was some reason to investigate hydroxychloroquine. There were some theoretical justifications for investigating it, and of course, there was the unbelievable enthusiasm expressed by some people in high positions for this drug. And then, as new information came out, basically any original optimism just faded. However, there are still so many people out there who harken back to the original, optimism? How do we explain this? How is it that they are not convinced by the data has emerged now over several months that hydroxychloroquine just doesn't do anything?
All right, well, let me respond in two different ways. First, I want to just talk a little bit about the science and statistics of clinical trials. So anytime you run a clinical trial, you're trying to estimate whether a drug is effective. Think about it, if you're a research sponsor or a researcher and you get an amazingly large effect, you're probably going to quickly try to announce those results because they're exciting, they're potentially impactful, more quickly than if you get a result that is ambiguous or if you get a result that's null, right? The problem is that anytime you do a clinical trial, there are two different factors that can influence the measured effect that you get in a clinical trial. One of them is the real effect of the drug, and that's ideally what we're measuring in the clinical trial. And the other is random variation, which we can't do anything about. I mean, you just got unlucky. Sometimes we just get unlucky. You run a clinical trial, you see an effect, and it's not a real effect. It's just we just rolled the dice accident, and the wrong number came up. And so if you put those 2 points that I made together, what that means is that oftentimes when you get really big effects early on in a clinical trial, as you continue to run that clinical trial, those effects will start to regress. They'll get a little bit smaller with time. So that's probably, that might very well be what happened with remdesivir. You got these really big and exciting effects. In the United States, there were big press releases about this being really, really promising. They were small clinical trials, and small clinical trials are more buffeted by random variation. But that when remdesivir was put into a much, much larger trial, those big effects, or really kind of more like moderate effects, regressed back towards a non-effect. And so that's something we see again and again. And I think that, with these vaccines, it's not unreasonable to anticipate there'll be some regression of an effect, that maybe not 95%, maybe it'll go down to 90% or 85 or something like that. That's just based on the historical record. Now, to the second part of your question, why do people persevere in believing that a treatment like hydroxychloroquine, which has now been demonstrably shown to be ineffective in preventing or controlling, COVID-19. Why do people persevere in believing that? That is a question about human psychology, and I think that to really kind of answer that question, you'd really need to talk to people who understand, human psychology and cognitive psychology. And I think you would also probably need to talk to people who understand the way information flows in our society. So right before this conversation, I was actually reading a New York Times article about what they call super spreader, misinformation events. Not all misinformation spreads as rapidly as other misinformation, much in the same way that not everyone who has SARS-CoV-2 infection spreads the disease as rapidly to others. So information and viruses have a lot of similarity, and I think now there's emerging science about how it is that misinformation potentially spreads. So I think I can't answer your question. Unfortunately, I'm neither an expert in cognitive psychology nor in sort of information trafficking and flowing to give you a really good answer. But what I would say is, if we are going to have effective public health measures, having sophisticated social science, sociology, psychology to inform our public health policies is going to be paramount. It's not just about laboratory research and biology and molecular biology. It's about coupling that to really good understanding of how people understand science and how people make decisions about it.
Well, one problem we have in this whole situation is that besides science, where there actually is some smidgen of truth to it that then gets blown out of proportion, like with remdesivir, etc., there are also all kinds of recommendations which have no basis whatsoever. So, for example, we saw last week Ben Carson, who came down with COVID-19. He's the secretary. What was he of
housing?
HUD.
I think.
Yeah.
Yeah.
Housing and urban development.
Something that he has zero expertise in anyway. He's a former neurosurgeon.
But he knows a lot about pyramids.
Yes, he knows a lot about pyramids. Yeah. We'll get into Ben Carson's lack of knowledge about anything another time. But he said that he was cured of COVID-19 by taking oleandrin, which is total nonsense. And this was basically promoted by the pillow salesman, who has made a name for himself on TV selling pillows. And when someone of the stature of Ben Carson talks publicly about this, then of course a lot of people are going to jump on that bandwagon. When here there is no science behind it whatsoever. And Jonathan, of course, I mean, you spend your life looking into false claims like this. What do you say?
Well, I mean, part of that story of oleandrin, but also of hydroxychloroquine, part of the reason I think, from what I've seen, from what I've heard, why it has such a sustained, like, lasting power in the minds of certain people is that it is perceived as being this cheap thing that either cannot be patented or is now off patent. And so pharmaceutical companies can't make a lot of profits off of them. Therefore, they're not interested in pursuing that, or they're going to suppress the truth about how these things really work because, they can't make any money off of that. And so if you have a sort of a core distrust of pharmaceutical companies, that becomes a narrative that is very appealing to you. It's this idea that, well, there's this very, very cheap drug, hydroxychloroquine. And I've heard from knowledgeable people, from even the president of our country, I've heard that it works. But of course, this is being suppressed by pharmaceutical companies because even though they're the ones selling the drug, it doesn't make enough money. They want to sell you a new drug that will make them a lot more money. So that's an appealing narrative.
Right. And of course, we see that argument now with the vaccines too, that they're coming out with the vaccines without sufficient evidence because they're going to make a whole lot of money on it, which of course is nonsense. Dr. Kimmelman, I'll distinguish between the
yes.
two. One of the piece of data that has struck me with all three of these studies is that the number of cases is significantly lower than what one would expect, because when we see all the data from what is going on in the U.S., where you're getting positive, like in North Dakota now, they're getting 80% positive cases all testing. In all of these studies, the positive cases are very, very low. Now, why I think that the people who sign up for these studies, who are subjects, are people who are health conscious anyway. You're not going to get your seniors with comorbidities who are going to be the subjects here. So just the fact that we are looking at a population here that probably wears masks and pays attention to everything that they're supposed to pay attention to means that we don't really have, in all these studies, we don't really have a cross-section of the population. Well, what do you think of that?
Yeah, well, I mean, I think you're right. I have a couple different thoughts. So first of all, this is kind of a recurring theme with COVID trials is, a lot of these trials are depending on cases. They're what we call in epidemiology event-based trials. And, a lot of them are having a harder time than you would think accumulating enough events to be able to do the kinds of analyses that you would need to be confident that you can conclude something is safe and effective. So that's the first thing I want to say. The second thing I want to say is I agree that, I haven't done the math, I'm sure others have, but we're talking about a fairly small number of cases, a 30,000, trial of 30,000 people, only about 150 or so people who have actually, developed SARS-CoV-2 or COVID-19 rather. And so I think that, this is another reason why I think we ought to be a little bit cautious about overinterpreting that result, because in fact, we expect there'll be a lot more cases. And so who knows, what the effect size will be once we have more cases. So that's a second point. The third point is about what we often call external validity. So the idea is that when you do a clinical trial, the ideal is that you are sampling a population that is identical to or exchangeable with the population you want to use the treatment in. So that if you see efficacy in that population, you can infer that you'll see the exact same efficacy in the unsampled population, the people who did not participate in the clinical trials. It is well known in medicine that the people who participate in clinical trials are different than the people who don't. One way that they're different is that to participate in a clinical trial, you typically have to give informed consent. Some people don't give informed consent. They don't want to go into a trial. And some of those people have different characteristics. Either biologically they're different, or they behave differently, they have different exposures. And that means that when we get an effect in a clinical trial, we don't always know the extent to which that will generalize to the unsampled population. In this case, I don't know, I haven't seen any data, and I think this is one of the things that press releases will not allow you to assess. What you would need is baseline data on the characteristics of the patients or the volunteers who were in those studies, so that you can compare that with the demography of people who generally, are out in the general population. And, if you see a lot of disagreement or discordance between the baseline characteristics and the real world, you have greater reason to think there may be differences. Now, I can speculate that I think it sounds plausible to me that the kinds of people who participate in these early trials are people who are probably more committed to practicing, safe social distancing, people who are probably wearing masks, et cetera. So you might expect lower case numbers in that population. And again, I can speculate, I'm not saying this is true, but I can speculate that you might expect to see a different effect in that population than in an unsampled population. So for example, it may be that the people participating in the vaccine studies have a lower dose of exposure to SARS-CoV-2 than people who are not participating. So the question is the vaccine as effective in people who get massive exposure to SARS-CoV-2? Those will be the kinds of questions we'll need to answer as we continue to follow these vaccines, as we do other vaccine trials, and as we pursue what's called pharmacovigilance, as we pursue evaluation of the product after the clinical trial. Now, there's something I want to really emphasize for listeners here. Some people, when they hear me say that there's a lot of uncertainty right now about these vaccines, what they may be hearing me say is, oh my God, this is really scary. We don't really know for sure about the safety. We don't really know for sure about the efficacy. And my response is, welcome to medicine. We all the time, drugs get approved and are used in medicine for which we don't have as much information as we would like about the safety and efficacy of a product. If you're a cancer patient and you go to your doctor and your oncologist recommends a certain treatment, that doesn't mean that the physician knows everything, that the drug is perfectly safe and the physician knows 100% it's going to be effective in you. What that means is that the physician, presumably if he or she has done her job properly, has reason to think that overall, given what we know about this drug and what we don't know about it, overall, it's better to recommend using this than not. And the same thing is true for vaccines. Yes, we have uncertainty about vaccines, much in the same way we have uncertainty about cancer medications when you're a cancer patient. But based on the evidence that we've seen, we have reason to think that the benefits far exceed the risks of undertaking these interventions.
Benefit risk analysis is the key.
It's also very important to point out that the alternative to getting the vaccine is not risk-free. Because we often think of this as like, well, I can take this thing that is risky, or I cannot take it and then remain safe. But we have this virus spreading around, and that is not a safe situation. So there are risks to both of these sides.
Also, right. Yeah. I really want to underscore that. It is really important. So that's what again in our field we call this counterfactuality. What would happen if you didn't do something? So in a way, that's what a clinical trial is doing. You got one group that's getting the placebo. That's the counterfactual. Those are the people who are doing what we would normally do, versus the people who are getting the treatment. And in this case, the counterfactual, the effect of not getting the vaccine for these these two trials at least, is that you have a much, much more elevated risk of eventually developing COVID-19. And again, think about this in the context of cancer. Yes, you can refuse the cancer drug because there's still some uncertainties about safety, and we don't know that it's going to work in you in particular with 100% certainty. But what's the alternative? We know what the course is for a person with cancer. We know that cancer patients left untreated will eventually advance, and they'll advance more rapidly probably than if they get the drug. So you make a good choice given what you know about the risk and benefit.
Something else we should point out about all these three trials is that they don't give any evidence at all for asymptomatic transmission, which is one of the most important things that we worry about, because I mean, let's face it, these days you meet anybody and you're suspicious of them, right? You suspect anyone can transmit it, which is a possibility. I mean, it's a real possibility. But this trial none of these trials tell us anything about that, because that in fact would be, I think, a very difficult thing to do, because you probably have to be testing these, well, now altogether in these trials, 70 or 80,000 people, you have to be testing them every day to see whether or not they are positive. Because as we know, you can be positive one day, negative the next day, then positive again. It's almost undoable.
Yeah. So two things. First of all, I believe the AstraZeneca study is actually monitoring that. I'm not aware that they've gotten.
I believe so as well. Yeah.
So, but to my knowledge, the Moderna and the Pfizer have not measured that. But I think the important point to underscore here is that there is a lot that we will learn from these clinical trials, but there's a lot they will leave unresolved. And so it's important to recognize that the task of doing systematic evaluation of these vaccines doesn't end the day that the vaccines get approved. It is going to have to continue. We're going to have to continue doing clinical trials, collecting information in registries, et cetera, so that we can continue to reduce our uncertainty around any number of questions, including the question about transmission. Now, I think that I want to underscore there that companies often won't do this out of the goodness of their heart or the generosity. Companies only do this insofar as regulators like Health Canada and here, or in the FDA, make them do it. And so, it's our task, I believe, as taxpayers, to make sure that we have a really good, competent, and effective regulatory apparatus that is going to keep on those companies to gather that information.
Okay, let's get into an interesting ethical question here. We have the AstraZeneca vaccine, which is according to them, going to be about $3 a dose. And the Moderna and the Pfizer are $20 to $30. So we have basically a tenfold difference in cost. Now, let's assume that the stats that they have given us so far are correct. And let's say that the AstraZeneca is 70% effective and the other two are 95% effective. So there's a decision to be made here, right? Are you going to go with the cheaper vaccine, which is actually going to be more easily distributed also because you don't need the ultra low temperatures? You're going to go with the 70% cheaper vaccine or the 95% more expensive and more difficult to deploy vaccine?
Yeah, so what you're describing is perfect example of what we spend our time thinking about in bioethics. So you've got these two sort of competing priorities, access, in terms of cost, and effectiveness. And there's going to be other considerations like storage, for example. Will we be able to deliver these vaccines in remote parts of Canada? And are there particular populations who are either, especially at risk, or to whom we owe our healthcare priorities, for example, disadvantaged populations. So there are any number of different factors arrayed around the question of which vaccines we're going to prioritize, et cetera. And a lot of what we spend our time doing, in bioethics is thinking about what different factors ought to be considered and how they ought to be weighted with each other. And I would say in this case, ideally what you'd want is some kind of a formalized, what's called a decision analysis framework, where basically you can mathematically model, if you sort of set the different elements, the different parameters, fairness, cost, access, disadvantaged populations, you can essentially kind of run mathematical models to look at what the value for society will be of different kinds of systems for prioritizing different vaccines. And I hope that public health authorities are undertaking those kinds of formal decision analyses.
Isn't part of the question moot, though, because many doses were pre-purchased by the government in advance before data on efficacy was even known, right?
So we're getting close to the edge of where I feel like my competency is. So I'm gonna answer that question, and then I'm gonna stop.
Sure.
To be sure, there have been some advanced purchases. I'm not sure whether the advanced purchases are sufficient to cover, the entire Canadian population. So I would imagine that even with those advanced purchases, there's going to have to still be further investment and purchase of vaccines. And even with the ones that we have, we're still going to have to make decisions about who's getting which ones and how we're using them. So I'm not sure it's
because as far as we go here in Canada, yes, there have been commitments for purchasing, but all of the companies have said that they will fulfill their own needs first. So both Moderna and Pfizer will first distribute in the US before anywhere else, and the AstraZeneca and Oxford in the UK and the US. So we're down the list until we even think about whether we want to take the vaccine or not. But when we
it may be, again, this is not something I understand well. I guess I feel very grateful to live in Canada, where we have an effective public health system. We may complain and sometimes wish our healthcare system, was better. But looking at our neighbors to the south, at the way the COVID-19, the way testing has unfolded, management, et cetera, it's been an absolute debacle. And so, while it may very well be true that there may be earlier access and purchase of vaccines down there, whether in fact the distribution and implementation of the vaccine will be faster than Canada, I think there are good reasons not to worry about that.
So I guess what you're saying is let the Americans be the guinea pigs, and we'll see what happens.
That's maybe partly, but I'm also saying that to get this vaccine to people, you need an effective healthcare system and public health system. And if you look at how the utter debacle of testing, much less the management has been, of SARS-CoV-2 in the U.S. has been, I think I don't think their distribution systems. We don't have reason to think the distribution system is going to work
right,
well.
Right, and also, I mean, let's face it. So far, none of these vaccines have been approved. So we're still waiting on that one to see if the emergency use application actually goes through, because FDA, of course, is going to have all the data, not only the press release that we've seen.
That's right, and I guess I'll say one last point, which is that the fact that at least 2 of the vaccines on preliminary look, has such potent efficacy and such safety. Now, again, there's stuff that we don't know, but so far, the fact that 2, and that a 3rd one looks pretty promising. There's some confusing stuff in the data, but even the lowest estimate from that trial, 60% or 70% is not bad. Suggests that it might not be that hard to develop vaccines for this particular virus. Now, there are at least 8 other candidates that are in phase 3 development. So, and there's also products that are in development here in Canada. So, I think there's reason to expect, to be hopeful, and to be optimistic that it may not be difficult to have a diversity of, effective virus platforms, which bodes well for being able to get good distribution around the world.
I think even if the vaccine does get approved, emergency approval, and they start deploying it, I think we still have to understand that we'll have to keep with the physical distancing and the masks for quite a while. This is not going to be that as soon as the vaccine starts to roll out, then we can just relax. And I think that is a concern, because I think a lot of people are going to look at it like that. Yeah, thank goodness the vaccine is here, and now we can go about our usual life.
Another concern also is that a lot of these vaccines require 2 doses that are spread a few weeks apart. And the side effect profile that we're seeing being reported in those press releases and these early sort of interim analyses can involve fever for a significant portion of the people who get it, can involve muscle aches. So you kind of feel like you have the flu for a day or 2. And so one of the worries is that how many of the people who experience these side effects will line up to get the second shot? Because if
Right.
they don't, that also will have an impact on how well they are protected and how well the society as a whole is protected.
Well, one issue with the two-dose vaccine is that in some cases, people may actually develop an immune reaction to the virus itself, the vector itself. And the Russians now are really playing this up, because their vaccine, the Sputnik vaccine, and I must give them credit, it's a very clever name to have come up with. It's very good. That is also a two-dose vaccine, but the second dose is not the same adenovirus as the first one.
Right.
So they're trying to circumvent the problem of actually developing an immune reaction to the virus. Again, who knows? We'll see whether or not this is true or not. Jonathan, Dr. Kimmelman, what do you think about the Russian studies? Have you seen any data about that? Because I haven't. I mean, they just say that they're testing it on 40,000 people, but they haven't released anything. So who knows? Have you seen anything?
I haven't. I guess you can count me as a bit of a skeptic about the medical research that's coming out of Russia when I look across other areas. So, for example, stem cell research. There are certain countries that tend to harbor kind of rogue research actors that don't have really effective kind of regulatory apparatuses set up. And so you see, recurrently reports coming out of different places, and Russia's one of the countries that I worry a little bit about in terms of having a good regulatory apparatus, having a scientific system that has not been politicized, et cetera. So I guess you can count me as a bit of a skeptic. Doesn't mean for sure that Sputnik, what they're calling Sputnik, which I think is actually a terrible name. I think it's a horrible name for a vaccine, in my opinion. All the kind of Cold War connotations, all the kind of jingoism is just exactly what we don't want with vaccines. But nevertheless, if it may very well be the case that Russia has a very, very effective vaccine. I'll wait to see the data, but I start with a bit of skepticism.
Well, I think the reason they chose the name Sputnik, of course, is because it's a breakthrough. It's a new development, like the original Sputnik was. And remember how scared we were when that Sputnik passed over North America and gave out that beeping signal saying that we're here. And of course, the message was that if we can put that Sputnik up there, we can also launch a ballistic missile,
That's true. Joe, you're probably the only one on this call that was alive then, too.
Yes, I was, and I certainly remember that. I mean, that was quite an era. Anyway, we're kind of running out of time here. So let me put both of you on the spot. Suppose the vaccine...
The spot or the Sputnik?
Suppose the virus, one of these vaccines gets approved. FDA approves it, Health Canada approves it, whatever, and rolls out, and you can get it. Would you get it? Would you be first in line?
I'll go first to let the other Jonathan on the call think about this some more. I mean, first of all, I will not be among the first ones to get vaccinated because I'm not in an at-risk group. So I'll be, if I get vaccinated this summer, it's going to be pretty early on. I will look at the data itself, and I will consult with other people, statisticians and medical doctors and people who are experts at this to get their perspective on this. If
FDA approves it, that is what has happened. They have consulted with all of the experts.
Well, to me, that's not sufficient in these exceptional circumstances, and given the politicization of a lot of the agencies in the U.S., unfortunately under Trump, I would like to get expert eyes on the raw data, on the data that is available. But I mean, if it does look good and if it does look safe and effective, yes, I will definitely get the vaccine.
Okay, the other Jonathan.
Yeah, I don't have much to add. I think Jonathan sort of hit on the key points. So I think, FDA has really taken a beating throughout COVID-19. They botched so many decisions. I mean, I've been critical of FDA, before COVID, but I always sort of clung to the idea that this was at least, still a functional agency. And I think throughout this, I've begun to have, my doubts have actually, heightened. Having said that, the vaccine branch, I mean, it's important, these administrative structures have different branches, and what I've seen from the vaccine branch, the biologics branch, has been much more comforting. I have much greater confidence in that element. Ultimately, here in Canada, so Jonathan's right, the other Jonathan, that we're probably not first in line to get a vaccine. There'll probably be other people who are at higher risk, healthcare workers, et cetera. I do have a confidence in Health Canada's reviewers and approval system, and I like Jonathan, will want to see what people are saying. I want to look at the data myself and see what other statisticians who I trust are saying about the data. But that combined with if that checks out, combined with Health Canada approval, I'd gladly get a vaccination and be able to travel to Germany to give my talks and like I normally do.
Okay, in the meantime, we will put on our masks, wash our hands, right? Keep the physical distancing, not the social distancing. We don't want to be socially isolated. Big difference between physical and social isolation. So thanks to both Jonathans. I think we had a nice, lively discussion here. I think we addressed a lot of issues. We haven't solved any problems, but I think we've shed some light on some of the confusion, and there is that little glimmer of hope at the end of the tunnel because of the vaccines. So guys, thanks very much. And maybe Dr. Jonathan will check back with you again in a few months when we have some more info on just what is being approved, and by then probably we'll have some other vaccines that have appeared as well. All right, and thanks to everyone for listening, and we'll be back with you in a couple of weeks.
Thanks for having me.
Bye now.